share screen.
En dan van ek maar weer op my rekenaar.
Sal dit
Net weer
waardeer?
as daar vrae is.
Sal dit waardeer?
100%. Seker. Okay, share.
Hierdie ding record nou outomaties, nee.
Ja, ek het nog outomaties
Goed,
gesien.
so nee, daar was 'n vraag gewees of het record, so.
Ek het nou julle webinar van gister en ek het julle attendance register afgetrek in die video. So daar was 'n klare request van een van die leerders wat nie kon bywoon nie vir die chemie ene. En miskien kom dan nog. So dit het eintlik baie goed gegaan gister in die webinars.
Goed.
Ek ben bly met dit is.
Waar is geshare? Hoekom is daai nou daar? Hoe is daai omtrek? Yes, Johan.
Yes.
Nee, alright. Goed te sien, goed om te sien.
Ek het die ding op die system gelaai. Kan dis wat daarvan af hardloop.
Jy kan dit probeer doen. Kom ek kyk net gou of ek hierdie kan uitsorteer. Dat is hier. Wys uit daai kant reg. En nou moet ek net die sharing reg doen.
Maar jy gaan net op een plek wys.
Ja, hy wys net op een plek, ja. Stop sharing. Share. Daai screen. Share dit over. Okay, daai lyk reg. Okay, okay, nou moet jy vir my sê wat jy het gesê.
O nee, ek gaan met anders die sien alternatief.
Het jy stokkie?
Ja. Okay. Whatever die maklikste is. Ek het present wat jy het ook.
Ek het ook 'n presenter as jy. Kom ons doen dit so. Waarom is hom verloor?
100%.
Perfect. Alright. Sit ons maar net op my skerm dat se mense join. Easy ook.
So. 100%.
Perfect.
Hoi.
Dan goed om te zien. Ik heb een notebook. Ik weet niet of je weet wat die voor.
Dis die maklikste. As ons dit op die rekenaar sit, dan werk of ergens, maar dis we can make a plan.
Pieter Neethling.
Ik kan ze nog een nooit. Ik heb ook van graphics wat er het en zo. Hopelijk is die computer beefy.
Beefy genoeg.
Ja.
Sal maar moet kyk.
Nog verder man ook aangestoot.
Ja. Almal hier. Great.
Man is opgewonde.
Ja. Vroeg aan die gang, dis reg.
Dus ik vond.
Okay, op wakker vir. Kristian, kan ons dit van die rekenaar af hardloop of moet ons van jou laptop af?
Ek het dit nou is dit beter om dit vanaf die rekenaar.
Die voordeel hier is dan share ons met die teams aanlyn as met jou ook van jou te rekenaar of met die team share. Dis hier.
ek het nie 'n ding wat hier inpas nie.
Dit kan ek vir jou kry.
Maar ek het dit op my shared drive.
Ja, dan kan jy dit
Of
aftrek.
ek kan hom met van my Dropbox vir jou stuur.
Maar ek kry vir jou 'n konverter. Ek het 'n konverter in my kantoor.
Ek gaan
Easy
dit nou eers doen.
Ja, waarby?
Nie alles kan werk nie, maar die meeste van dit.
So ek sê anderster,
Ja.
nee, as jy geregistreer het vir hierdie praatjie, dan het jy die Teams link daar gesit. Okay. Want dis die ding, jy moet dan nou share ook. So dis die okay, ek kry vir die konverter. Hier jy.
En baie so,
O, hy die stokkie nog.
I'm still standing. If I had to do karaoke, I would do that stuff. I'm still standing. It's funny, I've actually been between that. I'm trying to build.
ek het dit convert van die tools. Dit lyk daarop. Tenminste as ek die PowerPoint op die Mac probeer.
Ja, dis 'n goeie kans dan.
Ja.
Maar ons Apples met dat al.
Okay.
There we go. Press me aan daar op.
The one last thing about
Take it from there.
forward to the right message.
Wat enige voorkeur en volgorde?
Ek dink op die soos op die program is fyn met my.
That's the events you have the staff. Yes.
Now you'll use the who's start. Yeah.
That must be room. Yeah.
Hopelijk misschien kan ik met toets.
Ja, as kyk vinnig kyk. As jy wil.
It was karaoke.
Swanson.
But if I had to choose a song, that would be what the stuff is.
Dis jy omneem kan. Ja. Ja, right, eers dan die woord. Okay. Wil jy iets?
Ik ga net vir jou die check.
Thank you.
Hoe lang is weet nie hoe dit werk.
So even
Op hom klik dan kan hy.
O ja, okay. En ek doen automatisch te
Want dit gaan outomaties gaan dit.
Ja,
En never mind hoe jy dit stel, dit gaan altyd.
okay. Is het automatisch te doen? Okay. Die movies was just zo'n vroeger jaar was die resolutie van okay.
Minimize hom sommer net.
Okay.
Dan gaan ek net
Dit is kyk.
PC se pointer is op stop. Wat is stop? Nou, is laat ik. Okay, good evening, everybody. I suggest we start. My name is Pieter Neethling, and I'll be the MC for this evening. So first of all, welcome to all of you for what will be our 9th annual Science Behind the Nobel Prizes lecture series. So I think it's something the Science Faculty can be proud of, that it's been continuing for 9 years and still going strong. So thank you all for attending. So first of all, just how we will approach this evening. First of all, I've got to thank Mythics, who is sponsoring the snacks and drinks that you'll be enjoying afterwards. So before I forget, we've got to thank them for financial contribution. And then just the program for the evening. So we'll start with the Nobel Prize in Physics, presented by Kristian Müller-Nedebock. I'll introduce him first, followed by Professor Johan Burger for the Nobel Prize in Medicine, before we move on to Professor Hugh Patterton for the Nobel Prize in Chemistry. Each presentation will be hopefully about 20 minutes long, after which there'll be time for questions before we move out. So that's about it. So I think without too much further ado, let me introduce Professor Kristian Müller-Nedebock. He obtained his PhD from Cambridge and is currently heads the Physics in Biology group at Stellenbosch University. He works on polymer and filament networks, active systems, and physics of biological structures at cellular and subcellular length scales. And this work is mainly theoretical, but of course he does it in collaboration with experimental physicists and other theorists. And he will, of course, be talking about the Nobel Prize in Physics, which this year was awarded to John Hopfield and Geoffrey Hinton for foundational discoveries and inventions that enable machine learning with artificial neural networks. And I'll allow him to tell you all about that. Yeah.
Thank you very much for coming. No, just put the wrong. Should go to the start. Oops. So, okay. So if you've seen me give some talks before, you might recognize some of the images, but you'll see that the connection is not tenuous at all. I also have to tell you that, as you heard, I work in the physics of biological systems, which means I'm not an AI expert in any way. There are probably many sitting in the room, but I'll speak about some of the physics ideas that lead to some of the perhaps machine learning ideas. So, as you already heard from Peter, the Nobel Prize this year is being awarded to John Hopfield and to Jeffrey Peterson from Princeton and the University of Toronto, respectively, for foundational discoveries and inventions that enable machine learning with artificial networks. So, what does that mean, at least for me, when I think about networks and the simplest kind of physics-based ideas that might be behind this? Interestingly, a few years ago, I stood here and gave another lecture on the Nobel Prize, and at one stage, I found a slide in that talk, which part of it had to do with spin glass systems. And I had a slide on Hopfield networks. So if you recall, the name Hopfield is one of the laureates, and that's the first thing that I'll be speaking about. So here are the two sort of seminal papers by these two authors lying on my desk, and mainly drawn from one in the Proceedings of the National Academy of Sciences in the USA from 1982, and another one a few years later by Hinton and co-workers, but there are many others, of course, on a learning algorithm to Boltzmann machines. The basic idea is that brains in some or other way are biological machines that are very good at identifying and processing patterns. But there are also other biological machines that identify and process patterns or that follow patterns, for example, proteins that know how to fold very quickly into a certain configuration. So how do physicists, or perhaps how did John Hopfield and some other physicists at some stage think about neurons? Well, physicists, and if you're a third-year physics student at Stellenbosch, you will know about this, learn about magnets. Why do we learn about magnets with certain kind of spins? Because they happen to be the simplest systems. Physicists are relatively simple-minded when it comes to thinking about relatively simple-minded when they try to introduce models to think about certain systems. So the spin system that our third-year physics, statistics, or physics students would learn about is one that has just an up or a downward pointing spin. And the systems become interesting when the spins start talking with each other, when there's some kind of interaction that costs a certain amount of energy. Depending on what kind of material these spins sit, sometimes it's energetically favorable. They point in the same direction. They can be alongside each other like that, one above the other like this. And perhaps it's more energetically costly to point in opposite directions. So the important message is these are some other physics systems that just have two possible states, up and down. Call them -1, +1, or you could call it 0, 0. The only thing that really counts is that they're two states, and that these states can talk to each other through energy. Okay. So now if we can, there. So energy, of course, is one of these basic currencies. And if you cool a system down very far, the kind of energy the system itself wants to be at is the lowest energy. So here's some or other block in some or other landscape, and perhaps there's a gravitational force pulling it downwards. Oops. And here it costs more energy to have the block on the top of the table, slightly less to have it on the bench over here, but certainly putting it downstairs is the cheapest in terms of energy. But that's not the whole story in most cases, because all of the physics systems that we usually have, and you'll hear why this is relevant, is that sometimes there's a competition with energy. Whereas the energy wants something to sit as low as possible in this landscape. There are, of course, other forces that wanted to explore the landscape. And this other thing that wanted to explore the landscape we can call entropy, it wants all of the freedom as possible. The particle wants to sit on top of the hill over here and there. And so we think about a quantity called free energy in certain cases, which is a kind of mathematical competition. The competition is through the minus sign between S, which is entropy, and E, which is energy. This F needs to be as small as possible, which means E must be as small as possible under the condition that S is also as big as possible. But you see, we multiply it by S. So if T is very big, S wins in making F as small as possible. If T is very small, E wins. So here's a kind of sort of basic equation of everything from the perspective of the statistical physicist. And it's this kind of idea, together with the fact that we're looking at a system that just has two possible states, that inspires how we think about a ferromagnet. So what happens is, if we have a system of spins here, so they're not just pointing down, but in various directions, it's simulation. At high temperatures, these arrows want to explore all the possible orientations that they can have. As you lower the temperature, at some point, the arrows start getting sort of some kind of coordination. There's some kind of unified sense of direction that they adapt. So the system, the magnet, once it's cooled, goes into an ordered state. And one can really just understand this through that simple equation that you want to make this free energy the same. So third-year physics students would also do a course in computational physics, and some of their images might look like this. At a certain point, they're trying to figure out, how do the islands of magnetization in such a magnet look, and here are some different temperatures, and you can see how, in various different ways, explore how these magnets sort of start getting ordered. So the important thing is spins with states, and they're talking to each other, and there's this kind of competition. The computational scheme that third-year physics students use in this is called a Monte Carlo simulation, or simulated annealing. Again, some kind of terminology, I'd like you just to not completely forget until later. So, how can one make this more complicated? Well, one of the things that was investigated sort of in the 1970s, 10 years before Hochfield's article was published, was this idea of a so-called spin glass. It's a variation of the previous models of these arrows talking with each other, but now the arrows don't all talk with each other with the same strength. That means there might be different energies associated with how the arrows want to be aligned. In particular cases, there's something that's called frustration. For example, here are three possible spins. But, for example, along the spin on the left with the one on the top on the right would want for energetic favorability to be aligned both in the same direction, but for some other reason, and we can certainly construct such materials, for example, copper and manganese in copper is one of these systems where you can find this. There might be a favorable reaction that the spin up here is energetically favorable with the spin down there, but then there could be some kind of incompatibility along the third axis. The interaction of here does not favor them to be in the same direction. This is called frustration, because you don't know how to fit the spins in order to completely satisfy, going to the lowest possible energy. So there are these frustrated states of physics that you have a very good definition of frustration. You don't know which way to go. So, as you can see over here, this kind of system consists of these spins. They're talking to each other, and because they're sort of slightly randomly diluted, there's an interaction that goes sometimes positive, sometimes negative. Distance, positive and negative, means they favor the same direction, but they favor opposite directions with different strengths. So now this magnet becomes a lot more complicated. And it turned out to be an almost 15-year
epic journey through complicated theoretical physics to understand what happens in these more complicated networks that might be formed, not networks, complicated spin systems that are formed in this way. And all kinds of strange and wonderful mathematics, and the Nobel Prize award was used in this way. But rather than reading all of the words in the slide, what happens is that certain frozen disorder needs to be taken into account, and that the magnet itself can settle down into a variety of different states, which are good free energy minimum states. So let's just sort of think about simple magnets in this somewhat cluttered diagram. Here's our system with spins that can point up or down. Okay. And they're all talking to each other. So there are 4 spins in this example. And with these arrows and the thickness of the arrows, I'm trying to indicate, favorability or unfavorability or strength of interaction. So how strongly mathematically is the energetic interaction between the spins along the diagonal, etc. You see, if there are 4 spins over here, and you make all of them talk with each other, there are 1, 2, 3, 4, 5, 6 different possible interactions. And they can be determined, and you can calculate an energy for this, which third-year physics students will know very well, is you sum up the spin and J, which tells you how strong the interaction is between spin I and spin J, and perhaps there's something external dependent on each spin, what we can call an external magnetic field acting on it. This simple energy, if you go and look in the physics library, has led to 20, 30 books exploring all different kinds of aspects of it. You can make it arbitrarily complicated, and it doesn't seem like a complicated thing because S can only be +1 or -1. J can be anything, H can be anything, but you can do a variety of things with it and get a really interesting landscape. So you don't have to remember this equation, but you'll see it again. The important thing is, once you look at this kind of system and you have got energy, you can start thinking, exploring the energy depending on all of the different ways in which the spins can point. Call that a configuration. And what that happens is you get something that's called an energy landscape. So as you move around this hilly countryside over here, certain states, state over here, which has a certain arrangement of spins, will have a different energy somewhere else. Okay, some energies will be higher, some will be lower. But remember, physical systems like, at least in one respect, finding the sort of the lowest parts of all of these values. So now let's think about our first memory network in the sense of John Hopfield. Say you have a 10 by 10 grid of spins. Some of the spins pointing up are yellow, the spins that are pointing down are blue. In the way that I've arranged them, it's some other pattern that looks almost like a letter S. Actually, I wrote it on this paper, I wrote it with my phone, then I rasterized it with mathematics. And that's what you get. Okay? So when I write X, and say X like this is associated with an energy on that landscape over here. So imagine you now take this physical system, so you put your finger on X, and you ask, can it fall into a valley where you actually have the nice X? So the question is, if you put approximate X somewhere, will the physics of this system eventually lead it to the value which is associated with the nice X, which means the set of spins, or we will call them neurons, has found the ideal configuration. It has identified this sort of, oh, this looks like an X to, well, this is the X that you found. Okay. So if the system can somehow slip down that hill into the right value. So that's Hopfield's neural network. And all of what one needs is, you have an initial state, and somehow the system needs to get to this target state, and probably slightly obscured by people in the meeting, if not, is the challenge. So if you want to go from an initial state in the system somehow to evolve to an ideal target state, so that it recalls a memory, given the rough sort of snapshot of what it's supposed to find, the challenge is, how does one set up this landscape? But that's not the only challenge, is how do you actually drive around on the landscape? What is the kind of physics that you need to do, or what kind of processes do you need to do, at least asking as a physicist, how you navigate this landscape? The two, of course, coupled to each other. So this is, what at least some of the sort of memory neural networks want to do on the basis of this Hopfield network. Coming back to this idea of a spin glass, is indeed this idea of the landscape. That is, after many, many years of really hard calculations, physicists figured out you wanted to find whether the system could find a local minimum, and they found with certain sets of spin glasses, there were really high barriers between these minima. So exceptionally high. So in a certain sense, it's ideal that you don't ever make an error in judgment in where you end up in the minimum. If only it were so. Okay. So here's some maths. In this Hopfield network. So I told you about these variables S that are only +1 and -1. They're neurons. +1 is one state of a neuron firing, -1 is the neuron is inactive. And so you describe your neural network, say what each neuron is doing at a certain time, +1, -1. You call this S, or you can call it the vector S. There's certain states of the neurons, so if the neurons are arranged as that perfect letter X, these are the target states or the memories that you want to recall. So in a network, of course, our brains, we want to store more than a single memory. So we need to store multiple memories. So there might be X, and we'll see perhaps a circle, perhaps a square, perhaps an arrow, or something like that. Hopfield says there's this energy, recall, this is exactly the same as previously. So Hopfield and Hansen predicts this. There we go. So he recalled his third year Stellenbosch University physics. That was a very important year, of course. But he recalled that from his undergraduate physics. And then he added something. How do we choose the interactions? So we have some freedom. He insisted on something that's called a head learning rule, which is essentially, using the target states you want and you multiply them in a certain way to start making these J's. Interactions, arrows between the up and down splits. And finally, to navigate the landscape, he included an up. So this is making the landscape. And to move around in the landscape, he had an update rule. So he calculated a bit of the energy, compared it to another bit of the energy, and he decided whether the neuron turns to a plus state to the minus state. Very, very simple, since these S's are just plus and minus ones, the Jij is, of course, slightly more complicated. So, this is what I did with Mathematica. Mathematica, yesterday, I decided I'm going to have some target states, 1, 2, 4. So my neurons, my spins are up and down. There's an X, there's a square, there's a circle, there's a filled circle, and there's a funny thing. Okay. So I decided this is hopefully going to somehow be imprinted on my system by this head learning rule, and I applied the head learning rule, and here's the 100 by 100 matrix that happens after you with the head learning rules, and the different colors just simply tell you how strong the connection between the 60th neuron and the 20th and the 30th neuron are. So sometimes the interaction is negative, sometimes it's positive. But that's what the head learning rule did. It just it's the map of every single interaction between a single pair of neurons. And I'm going to start priming my system, and here are my rasterized hand-drawn images. And to see if I rasterize them, if I put them into my neural net and let it evolve according to Hopfield's rule, will it somehow get close to these patterns? And so after a few hours of Mathematica, but not terribly expensive, you can see Not everything is animating. You have these different scenarios of different systems evolving. So in the first place, and the movie probably doesn't scroll in PowerPoint, Apple's thing. First system, I primed it as the X, and I let it evolve according to that rule. That doesn't look like an X, say where it ends up. Here, I put in the square, more or less. Could be a square, that's not very good. I put in a circle over here, that looks nice. The fill circle, that looks very nice. And here I put this funny arrow thing, and here, I can't even remember what I did in the previous one. Oh, yeah, just the curious arrow things in various ways, and saw how the system evolved. And you see the learning rule evolves in these videos, but the outcome is by no means reliable. Sometimes it seems to find, it seems to find quite easily the full circle or not. So one of the things one can do, one can just sort of adjust these J's, the J's that tell you how strongly it is coupled. And so this is what I did. I just retrained my network by showing an X. Retrain another network by showing it that final state, the thing that looked like an the thing that looked like an arrow twice, and it's evolving with time according to that rule, and it gets there. Okay, so by changing the weights in some other way, I didn't do it very cleverly, I can help the system to nudge ever closer to the kind of target. So I put in this very rough memory. And then if we're lucky, it evolves to the system. It evolves to the perfect X, and it evolves to this other shape. So all of this is a system moving around in this free energy space. Actually, the Hopfield model, as we saw it, is where the temperature is 0. You just go straight down the hill to the lowest energy scale. But what you see is this process is not right, and sometimes not reproducible because there's an element of randomness in there. And so, you know, how, and that's I suppose the question that learning people in some instances are trying to really optimize them. How do you reliably get targets think? So, what does one do if one is a third-year physics student to look at this? You go beyond Hopfield's rule and you add the temperature. And that's when you do the kind of simulation that brought about the ordering of this magnet and showed these really complicated diagrams about the ordering of the systems. And the real thing is, if you go and minimize the system landscape, you just roll down the hill. Imagine you've got a soccer ball and you're out of Kutzenberg and you roll it down and it ends up in the dam, but not the bottom rugby field. And you want it in the bottom rugby field. So you have to somehow kick it out of the dam. And that's what the temperature of the thermal environment does. And so if one wants to escape sort of minima that are local minima, but not the right minimum, you can use this idea of thermally shaping it, adding temperature, adding that entropy contribution, that disorder. And this is the idea of Boltzmann. And this seeded the idea of the second laureate, Hinton, one of his many ideas, but the one that's mentioned in the Nobel citation is where you use the idea of the Boltzmann dynamics, the fact that there is temperature, to actually help your algorithm find that right minimum. There are many different, very clever ways to do it. And of course, it also uses some kind of feedback that, as I did in making my X walk more recognizable to our system, you can retrain and always readjust those weights or the JIs, those interactions between points. And this one can do by getting about equilibrium statistical physics, at least in the initial states. So, I want to conclude and show that there are still physical, interesting neural networks. So recently, I chatted with some colleagues from the University of Pennsylvania, Andrea Liu and Greg Durian, and there is an interesting article published recently on University of Pennsylvania's website, where they've actually made a kind of network that can do certain actions, which is just a thing of resistors and simple electronic components coupled to each other. It's still noisy, it's still a completely physical operation, and it still does marvelous things. Perhaps it's not as intelligent as most of the machine learning that people at Stellenbosch are doing, but it's really very interesting that there's this physics basis to many of these interesting networks that can learn and do various actions that make use of an interesting way in which single components are coupled to many other components, and then collectively, if one can something, collectively go to a certain state, sequence of states that helps us to, for example, reproduce in memory. And that's all I have to say. Thank you.
Thank you very much, Kristian. As I mentioned at the start, we'll leave the questions till after all the speakers. So next up, we've got Professor Johan Burger. He received his PhD in microbiology from the University of Cape Town, but since 1991, he's been managing the research as principal investigator at the Agricultural Research Council, the CSIR, and Stellenbosch University, so many chairs. His current research is focused on various aspects of grapevine virology, with the ultimate aim of introducing virus resistance in grapevine cultivars. So let me switch. You can take that one, and I'll switch your. No cake. So. There we go. There we go.
All right, hello everybody, and from my side also, thank you very much for attending. It's almost Friday, and there are better things to do than sit and listen to talks that really got my head spinning. So total change of scenery. I've been asked, and thanks very much for the invite. Also, my second appearance as trying to explain the work that Nobel Prize winning scientists do, which is not the most trivial thing, but it's a real joy to be here nonetheless. So I'm going to be talking about the 2024 Nobel Prize for Physiology of Medicine by Victor Ambros and Gary Rufkin. I thought maybe before we get stuck into what these people did, I would just give a bit of a few interesting facts about the Nobel Prizes themselves. I guess, as most of you may know, there are 6 categories, 4 of which represent what we call the hard sciences. And 3 of those you will hear presentations of today, mine being the Physiology or Medicine Prize. What I did not know was that these various prizes are presented and administered by different institutes in either Sweden or Norway. And the Physiology and Medicine one is the only one that has been managed and presented by the Karolinska Institute in Stockholm. This particular prize was awarded for the first time in 1901 and has since been awarded 115 times to 229 Nobel Prize winners for this particular prize. Again, for Physiology or Medicine, the youngest person who ever got it was 32 years old. For the discovery of insulin, and the oldest, a guy from 87 for the discovery of human tumor-inducing viruses many years ago. Again, just a little bit of some fun statistics. If you look at, now we've got 4 disciplines, we also have economy here on the side, at the age at which the people, the average age at which the people did the work that led to the discovery and that led to them becoming Nobel laureates. And there we can see the different disciplines. The younger people are the likes of you, Kristian. the physics guys, and the oldest ones are the chemistry guys. So we cover the bases. So this is when the work was done. This is the gap between when the work was done and the final prize was awarded. And we see it's quite a few years. In my case, these guys did this work in 1993. They just got the prize 31 years later. So here again, the different categories and how long it takes after the work was done and when it finally got recognized and being awarded by a prize like this. And then the final one statistic, which I really do not understand at all, is the number of publications it took for these guys to get their prizes. Now, look at the numbers here. It simply doesn't make sense. If this was an indication, then I would have visited Stockholm a few times, and so would many of the people in this audience. And so I guess it's not the number, it's the what that you publish that will give you that prize. Right. So let's look at our winners, Victor Ambros and Gary Rifkin. And again, I tried to summarize their careers in a very tiny little nutshell, none of which is too important other than the red names that you see there. And all of those guys are Nobel Prize winners themselves. So I think the only thing one can deduce from this, these guys have come from serious pedigree. I mean, they have done their PhDs under Nobel laureates, they've done postdocs, and so on. So both of them have got a very, very, very solid upbringing in terms of the science that they find themselves in. I spoke to some friends a while ago, a week or so ago, after this became sort of weird, and they said, so what are you really going to talk about? I said, I'm going to talk about the Nobel Prize for Physiology and Medicine, and say, well, what did guys really do? And I said, well, it was for the discovery of microRNA and its role in post-transcriptional gene regulation. And this guy looked at me, the one of them, he says, you know, the only word in that entire sentence that I understand is the word discovery. Yeah. But that's actually the key word, because Nobel Prizes are all about discoveries and serious discoveries that impact on life. So this is exactly what these guys do. So I'm going to try for the rest of the night in my allocated time to try and pull this apart a little bit and explain what the various aspects mean. Now, in the interest of the non-biology scientist in the room, I'm going to give you and take you with me to my second year genetics, introductory molecular genetics course. And I apologize to the rest of the biologists that are sitting here that will be bored out of their minds. But I need to put things in context. So let's try. The human genome, i.e. our entire genetic code, comprise of almost, well, just over actually 3 billion of these genetic letters captured in DNA, right? We all know that. 300 billion is a lot. And it's surprising to think if a genome of that size, less than 2% of that actually is containing the codes or the recipes or the instructions for the proteins that will do the work and that will ultimately decide what the cell does eventually in a living system. Even more mind-blowing is the fact that our bodies contain approximately 36 trillion cells, and that is an even bigger number. And if you consider that every single one of those cells in its nucleus contain the entire complement of 3 billion genetic letters, it's kind of staggering statistics. If you take that, if you think that in our bodies 36 trillion, we cannot see a cell with the naked eye. Its nucleus is a fraction of the size of a cell. In that nucleus sits 3.2 billion of these letters. So if you take one cell in your body and you would string out that DNA approximately 2 meters tall. So very few people are taller than their genomes. I certainly am not. And the most staggering of it all, if you take 2,000 meters, you time it, my calculations are far simpler than yours. 2 meters by 36 trillion equates to twice the diameter of our solar system. in one person, the length of the DNA. It's a frightening thing to think of. Anyway, so one would wonder, why is it that all of these cells contain the full complement of the genetic code, and yet we have a situation where we have not only differently looking cells, but they do vastly different functions in our bodies. And the explanation for that is that not all of that 20,000 genes are switched on or activated or triggered, or whatever you want to call it, to work and to produce the proteins that are the working molecules in a living system. And that we call gene regulation. And this discovery has got everything to do with gene regulation. But before I go there, let's just talk again the conventional gene regulation system. Why do genes switch on? Why do they switch off? When does it happen and when does it not happen? And for that, I need to introduce you to something that is very classical. It's called the simple dogma of genetics, and it simply states DNA makes RNA, makes protein. And the protein, being the functional molecule, determine obviously the cell type and whatever a cell is doing. If we then think about this process of why certain genes are switched on and off, then we need to also think about the classical scenario of how genes are regulated. When are they switching on? And the process is illustrated clearly. Here we have, let's just call it a blue gene. Through a process of copying or transcribing, it's converted from double-stranded DNA to single-stranded messenger RNA. And the name messenger gives it away. It really does a temporary job. That, through a process, is translated from nucleotide sequence to amino acid sequence, of which are the building blocks of proteins, and they are the functional molecules, as I've said. And if we consider classical gene regulation, it happens by this process, copying DNA into RNA, this transcription process, by an essential binding of highly specified proteins called transcription factors that bind to certain areas in a gene to switch it on and off. So if that doesn't happen, the gene will not be transcribed, and this process will not follow flow, and the proteins won't be made. Hence, the gene will be silent. Now came our two friends who got this prize. They discovered a tiny, tiny little gene in the genome of a human. What they work in worms. I'll show you that just now. But in the same way that you will have a gene that will typically be transcribed, translated into a protein to do the job, this tiny little gene transcribed a microRNA, and the microRNA has got sequence complementarity that will cause it to bind to a messenger. And by doing that, it will inhibit the protein synthesis process, halting the expression of that protein, and obviously seriously influencing the regulation of a particular gene. Since the discovery of the first microRNA in 1993, more than 1,000 of these have now been identified in the human genome, which is quite significant. And I'm going to obviously talk about a few of these as we go. But before I do, I think I need to explain a little bit more about these baby RNAs, these tiny little microRNAs. What are these little things? And if you expect something spectacular and complicated, you're going to be seriously wrong. That is exactly the first microRNA. It's called Lin4. I'll explain a little bit more about it. The first microRNA that Ambrose and Rafkan discovered in 1993. They discovered it in a little worm. I'll say something about that as well. And as you can see, this actually is just a linear molecule of RNA, but its base pairing causes it to form this hammerhead or hairpin structure. The important part, this is a precursor of the functional molecule. The functional molecule looks like that. This thing is 77 nucleotides long. This is 21 nucleotides long. And this is the functional molecule. This is what is causing the gene regulation that has upturned the biological world with the discovery in 1993. This might look like a very trivial process. It's not. The process is quite complicated. I'm not going to run through it in detail, but it starts in the nucleus where you have a microRNA gene, which is transcribed, copied into RNA, forms this hairpin structure, and through a whole cascade of events impacting or making use of numerous enzymes and other proteins, it finally ends up being a single-stranded microRNA that binds certain proteins and have got rather serious impact on messenger RNAs and their ability to be translated into proteins. Right, so like I said, this all started in 1993 with a little worm called Physiological elegans. This is what it looks like. I'll say something about it more. And I'm going to run through this timeline in a little bit more detail now to explain exactly what these two guys did in the last couple of years to get to where we are today in our knowledge and the understanding of what all of this can do these days in terms of human health, plants, and the like. So Ambros and Rafkin actually met each other and did postdocs together in the laboratory of Robert Woolworth, another Nobel Prize winner, and they worked on development issues in Caenorhabditis, in this tiny little nematode worm for their respective postdoc project. Caenorhabditis elegans is a really well-known genetic model, and I guess that's the reason why they were working on this, because it's tiny, it's got a tiny genome, it's easy to grow, and it's got really well-defined differential genes, gene types. So that's what they were studying. Caenorhabditis also, I put this in red just to remind myself to tell you that when they interviewed Rafkin after he was awarded the Nobel Prize, he says the biggest achievement and the thing that makes him the proudest is that he could contribute to Caenorhabditis elegans achieving its 4th Nobel Prize. So it's the fourth time that this little organism was worked on and led to a discovery that yielded a Nobel Prize. And he was very chuffed with that idea. So as you can see, it's hardly a millimeter in length. You can only just see it with the naked eye. And it's a tiny, silly little thing. The two of them each work with a mutant line of this worm. And here you can see them. Here's the normal wild type. Here's the one which is called the Lin4 mutant. That is what Ambros worked with. And then the Lin14 mutant is what Rafkin worked with. And you can see this thing is a bit of a dwarfed phenotype. But they independently, in the same lab, work on these two mutant lines, trying to understand how this mutation impact on the phenotype that you get this kind of thing. They then split after their postdoc and established their own laboratories. And it's only after they split from each other and kept working on Lin4 versus Lin14 that they came together again and understood that there's a totally new way of gene regulation involved here. And that is what is ultimately led to the discovery. So independently, Ambros first, he was the guy that worked on the Lin4 gene. He suddenly realized that this gene is tiny. It's, in this case, Lin4 is 77 nucleotides long. That's minuscule. It cannot possibly encode for a protein. All of this is what he discovered. Rafkin, on the other hand, worked on Lin14, which is a proper gene. It encodes a protein. In fact, it encodes a transcription factor. And you will recall, I said a little bit earlier, transcription factors binding to promoter regions of genes are absolutely essential for the expression of that gene. If a transcription factor doesn't bind, the RNA polymerase II cannot get to this transcription initiation site, and there will be no messenger RNAs made, hence the gene will be inactive. Rifkin eventually cloned the entire Lin14 gene, sequenced it, and when him and Ambros compared sequences, they realized all of a sudden that Lin4 is a perfect match, or an almost perfect match, for Lin14 in terms of base pairing. And that made them think that, aha, maybe this has got something to do with the downregulation of this transcription factor, leading to those phenotypes of dwarfism that we've seen. So that was the speculation. They published independently 2 papers in exactly the same issue of the journal Cell at the time, in 1993. And then nothing happened. For a full 7 years, this was ignored by the science community. In an interview, Ruskin said the only comment that they got at the time was the scientific community said this must be a peculiar phenomenon, very unique to Caenorhabditis elegans. It will not be beyond this little organism. It took them 7 years before they discovered the second microRNA called LET7. And then the real impact of this discovery hit, because for the first time, this was not unique. Lin4, Lin14 are unique to Caenorhabditis. LET7 was then subsequently and very quickly discovered in most multicellular organisms, including us as humans. And that is what really caused a major outcry, and then people took them seriously. Now, many years later, we know, and I'll show you some examples just now, that improper microRNA expression leads to all kinds of horrible disorders and diseases. And it was also the founding biogenesis pathway for other small RNAs. And I personally think it is very apt and very relevant that they got the prize this year, because all of a sudden, the small RNA world is exploding. So microRNAs were the first to be discovered. Many others followed. And here's an example. Andy Fire and Craig Mello, they discovered something called siRNAs. Psi stands for small interfering RNAs, similar size, using the same biogenesis, that same cascade of events to generate a small single-strand RNA molecule that has got massive impact on gene expression. They got a Nobel Prize in 26. They only did their work in 98, so it took them like 8 years before they got a Nobel Prize for their discovery of siRNAs and the concept that they created. They called it RNAi, or RNA interference. But the small RNA world is a very, very hot topic in molecular genetics at the moment. Let's then look at the significance of their discovery in human health. This diagram gives you a bit of a rundown of where it also impacts. I'm going to try and summarize it very briefly in that it has been implicated in many very serious human diseases and disorders, including cardiovascular disease, autoimmune diseases, neurodegenerative diseases, and most cancers. Absolutely most cancers are impacted by irregular microRNA expression. The other very useful and topic and research environment these days is the discovery of biomarkers. A biomarker would be a molecule in your system that's easily detectable and would be associated with a particular disease. For instance, if you have most of them are proteins, which is associated, which is excreted in your bloodstream, for instance, that's associated with a particular cancer. That is a biomarker. It's much easier to detect the biomarker than the actual disease itself. So most of them, like I said, are protein-based, but these now can be used. MicroRNAs are available. They are detectable in human bodily fluids. So they are wonderful examples of biomarkers, and you can detect them in what we call a non-invasive liquid biopsy. You don't have to take a proper biopsy. You can have saliva, urine, blood, whatever you want, and you can, most of the time, you can pick up these things. The more exciting part in application is that of therapeutic uses of microRNAs against some of these diseases, against some of these cancers. And that is simply because people have now discovered and what they call antimers or antagonomers, which are anti-microRNAs. It would simply be a small RNA with complementarity to an RNA. They're also called microRNA sponges because they will absorb them and they will prevent them from doing what they do. And some of them are positive, some of them are negative. So if it's a bad microRNA, like an oncomer, that's the word, yeah, which are microRNAs involved with the development of cancer, if you can absorb them, you can prevent them from doing the bad stuff when they are expressed. Obviously, that is a potential cure or a treatment for a cancer. A real-life example that has gone commercial is a microRNA, a human microRNA called microRNA 122, which is expressed in human liver, and it is absolutely essential for the replication of hepatitis virus C. So if that microRNA is not expressed in the human liver, then hepatitis C cannot replicate. So they've created something, it's gone commercial, it's got a name called Moderator. It was the first anti-MIR drug going into clinical trials already in 2010, and today it's a drug treatment, it's a microRNA-based treatment against hepatitis C. Something that I just discovered when I prepared for this is really, really exciting, and I'm going to jump to the plant world just now. It's the application of plant-based microRNAs to downregulate or upregulate genes in human system. In other words, cross-kingdom. That is really very, very interesting concept. I'm a plant biologist, so you'll have to forgive me if I talk a little bit about plants and not all the time about medicine. And the reason is that there are far more work done since the Caenorhabditis work in plants than in humans or and animals, simply because plants are so much easier and you don't have ethical clearance and all those complicated issues. So it was very quickly realized that microRNAs in the plant environment impact on plant biological or biochemical pathways by impacting on transcription factors. And again, transcription factors being the key regulators of gene expression in eukaryotic organisms. And people have very quickly, and this is very complicated and very small, all it illustrates is the complexity and the number of microRNAs that have already been in the staple foods been discovered in response to most of these processes in plants, in development, in flowering, and everything that you can imagine here. The one that excites me is that of the responses against biotic, i.e. pathogens, or abiotic, i.e. physiological issues. A couple of years ago, and I'm very sad not to have a picture, I work in grapevine, and we did a project similar on trying to understand the microRNAs involved in when a phytoplasma infect grapevine. And in the process, we did RNA-seq, and we discovered a whole bunch of microRNAs in the grapevine genome, which is very exciting. But yeah, that was a while ago. What is being done in the plant world? Because of the experimentation, because of the fast progress in the plant side, people have started generating, synthesizing what they call artificial microRNAs or amiRNAs, which is a tool that is now being used very successfully to do all kinds of things in plants, to address abiotic stresses like drought and salinity, heat and cold, or biotic things like fungal, bacterial, and viral diseases, and even to, and very efficiently, to build in resistance to insects and nematodes. And the nice thing is this, if you express a microRNA against an essential gene of an insect, for instance, insects have got chitin in their exoskeletons. Chitin is produced by an enzyme called chitin synthase. If you synthesize an artificial microRNA that will target chitin synthase, and you express that in the plant, only insects that nibble and eat on that plant will ingest that microRNA, and it will inhibit their chitin synthase and they'll die. So it's a very clean and very precise technology, and very, very exciting. Similar technologies have been used to improve yield by impacting on physiological traits of particular crops, and then all other interesting contributions like seedlessness is highly desirable, increased shelf life is desirable, biofortification, i.e. improving the nutritional value, and also removing allergens and the like. In summary, I think it's fair to say that these tiny RNAs have got a profound impact and is profoundly important from a physiological perspective in life forms of higher organisms. These little microRNAs, in spite of being discovered only in 1993, have been at work for hundreds and hundreds of millions of years, impacting and allowing evolution to proceed. They play a key role in that. We have discovered that these cells and the tissues of multicellular higher organisms cannot develop normally if microRNAs are not involved, active. That is a fact. We have seen that if there's abnormal regulation as a result of microRNA expression, it leads to some serious diseases and disorders. And we've seen some of these applications nowadays in order to improve on human health and on sustainable food production. So I think Ambrose and Rafkin's discovery is really, and this tiny little innocent, hardly visible little worm has really revealed a new dimension of gene expression that hugely will impact on current and future life forms, I would think. With that, thank you. Are we going to do questions later?
Okay, now we'll leave the questions for the end. Perfect. All right. Yeah. Okay, so our last speaker, but if not the least speaker, is Professor Hugh Patterton, who obtained his PhD from the University of Cape Town and is the current director of the Center for Bioinformatics and Computational Biology, which he established at Stellenbosch University. His research focuses on the development of new bioinformatics methods and tools for the analysis of epigenomic data.
Right.
That one, and I think the rest is that. It's not, yeah, good. All right, so you, over to you. Thanks.
Okay, thanks very much. Again, thanks for attending and thanks for the invitation. It's always an honor. So I'm going to talk a little bit about the Nobel Prize this year in chemistry. So this is awarded to three people. Where's the, did I take the, is this the better pointer or?
That's the.
That's okay. Okay. Thanks. So David Baker, so he got his PhD from UCLA in biochemistry. He's currently working in the University of Washington. And then Dennis Hassabis, he was did a PhD at Cambridge in computer science. Interestingly enough, he was homeschooled and he obtained, apparently was a child prodigy in chess, and he obtained his A-levels at the age of 16, applied to Cambridge, and they told him, thanks very much for applying, but take a gap year, be too young. And so he went off and he spent a year working for a computer games company, and he basically wrote Frontier Park, and then went back to Cambridge and then studied and completed with PhD. And then John Junker, he also did an MPhil, I think, in theoretical chemistry at Cambridge, and then PhD in theoretical chemistry at the University of Chicago. Okay, so essentially, just to provide some context, and I think some of the background is actually painted by some of the other speakers already. So perhaps in terms of context, over here is just a scheme of a cell. In the cell, there are various functionalities, and the functionalities I'm just summarizing in terms of other growth, reproduction, detoxification, response to stimuli, etc. So there are many more functionalities in the cell, but this gives you an idea of what happens, so that you can think of it in terms of a system that has functionalities. Those functionalities are basically supported by a diverse series of chemical reactions. So sets of chemical reactions that basically underlie specific functionalities in the cell. So the problem is that if you look at these various chemical reactions, so this is basically simply an overview metabolic path of Saccharomyces cerevisiae. But if you look at the individual chemical reactions, there's an underlying problem. The problem is that a lot of these chemical reactions take a long time to basically occur. If you look at the half-life, if you look at the rate constant of the chemical reactions, some of them can actually take up to more than 1 billion years to get amino acid decarboxylation. So obviously, a living cell can't survive if you have to wait that long for individual chemical reactions constituting specific functionalities to occur. So you need to speed these things up. And the way to speed them up is basically using enzymes. And over here is just an example. So I'm zoomed in quite a bit, so you can't see the whole of the protein, the whole of the enzyme. Over here is essentially a substrate molecule. Over here is FAD, which is a cofactor in the molecule that becomes reduced, so a proton added to this. And then in the presence of oxygen, you have a hydroxyl group that's added to this carbon at this position of the substrate. So the reason I'm showing this to you is the way whereby the enzyme speeds up what is happening. It is taking this substrate molecule and it's actually sort of contorting it, bending it to resemble a transition state. So the transition state is the height of the transformation to a product molecule. The other thing, so this is just a single substrate that's using, but if it's something that where basically combines two compounds, then you'll have the enzyme molecule also orientating the two compounds in a way that would maximize the propensity of the electron clouds that need to participate in the formation of bonds to actually form those bonds. And then the third property here that's important is that in the space around this substrate, there are specific charges that are distributed. And some of these charges actually act as shepherds for the electron clouds. So basically, in the substrate molecule, they would basically herd the electrons to become more concentrated at some positions and depleted at other positions to actually allow the chemical reaction to occur. So the enzyme has very specific qualities that it's performing that is based on the structure of the enzyme that allows this particular chemical reaction to really speed up and for decarboxylation of an amino acid not to take more than 1 billion years. So they're here, and I think the image that Johan showed was much fancier than my image. But over here's the DNA molecule. And so basically in the DNA, you have the base pairs. So there are 4 different base pairs in DNA, G, A, T, and C. So if you have 4 base pairs, the number of bits that are carried is basically log 2 of 4, which is 2. So each base pair here represents 2 bits of information. So this is then transcribed to, over here is the mRNA that Johan referred to. And the mRNA is composed of codons, which are just triplets of nucleotides. And then there are tRNAs, and each tRNA has a specific anticodon, which is complementary to the various codons. And depending on the anticodon, it's charged with a specific amino acid. So if there's a specific codon over here, a specific tRNA with a specific amino acid will basically be complementary and bind to that codon, and then you'll have the addition of the amino acid associated with that charged tRNA to the growing peptide chain. So basically there's a ribosome, mRNA, you have the various tRNAs coming in, and they basically pair with the various codons. And this causes the translation of this particular sequence of codons into a sequence of amino acids. So you end up after this process with a long or large long string of amino acids or polypeptide. And then somehow this polypeptide folds into this really complex 3-dimensional structure. So the point here is that if you basically synthesize all these different polypeptides, they all fold into the same structure. So because you can crystallize them, so obviously this is a repetitive geometric unit. So it's not that it sort of assumes more or less the same structure. They're all identical, the same structure. So the only place that they can get, or where the protein gets the information to form this final structure, is from the sequence of amino acids itself in the polypeptide. And this was actually shown very conclusively by Anfinson from Johns Hopkins, who showed that if you denatured RNAs, I beg your pardon, if you denatured RNAs and you allowed it to reanneal, you can basically recover the activity of the enzyme. So in the absence of any external information input, the basically denatured protein will go back to its active form by allowing it to renature. Okay, so you can probably imagine that if you have polypeptide chains and you have protein structures, that there's likely to be a large number of these protein structures. Over here, we're showing you a sunburst type of representation of CATH, which is a database that stands for Class, Architecture, Topology, and Homologous Superfamily. And what I'd like you to see is that at the most fundamental level of class, there are basically 5 different classes. So alpha are proteins that are mostly alpha helix. Beta are proteins that are mostly beta strand or beta sheet. And then you get a mix of those, alpha beta, alpha helices plus beta strands, the arrows of the beta strands. You get a class with very few secondary structures, and then you get a class that's basically referred to as special, such as serum albumin, that is this unusual long series of alpha helices. So within this type of classification, you can see that there are a total of just over 5,800 superfamilies, which are the yellow the dial on the outside of the circle over here. And within those 5,841 superfamilies, there's over 100 million protein domains. So each protein domain isn't the same as a full protein. Some proteins are composed of multiple domains, but that gives you an indication of currently the total number of different protein folds that we are aware of. So it's an enormous number of protein folds. So the problem of that, so having so many protein folds comes in, if you look at the rate at which we're sequencing things. So this is basically taken from the NCBI website up to the end of last month. And by the end of last month, we actually have more than 3.7 billion sequences that's present in the database. If you look at the number of genomes, we have basically 2.5 million genomes. So 2.5 million genomes from different organisms that are present in the database. We have over 250,000 human genome sequences. So it feels like yesterday when we celebrated the fact that the first human genome was sequenced. We now have more than 250,000 of those. And in the UK and UK Biobank, we have almost another 250,000 human genomes. So we have an enormous number of these sequences available. So the problem is that if you have the sequence, that doesn't tell you much. You can identify the genes and you can perhaps identify some of the genes, but it's very difficult to sort of pull that through to the functionality. Remember the representation of the chemistry in the cell and pulling that through to the various catalytic steps and how they would work. So one of the things that one would try and do is take the genomes, identify the genes, and then try and identify the proteins that are associated with those individual genes in terms of being coded by those genes. But it's very clear that yes, you can sort of have this approach. It's not very accurate using BLAST or multiple alignment or homology modeling to get to the function. One of the easiest ways to try and get to the function to actually do the real structural solution of the protein by X-ray crystallography, that costs about $100,000. And it will take a postdoc at least a year to work on that if it's lucky that that particular protein crystallizes well. So with a large number of millions and millions of different protein folds, it's not really something that is feasible to try and solve these things by X-ray crystallography. So one of the questions that comes out is why are we interested in protein structure? So first of all, assign a function. We can also look at the evolutionary history of the protein. We can understand and study the reaction mechanism of particular enzymes, or we can basically use the structure to design new enzymes with new specificity. So there's a lot of value in actually understanding what the three-dimensional structure of a protein is. So because of that, there's this competition that has been running since 1994. It's called the Critical Assessment of Protein Structure Prediction, CASP. And the CASP competition will basically give the participants protein sequences. So this is simply a sequence of a protein with the individual amino acids represented by individual characters here, symbols. And the organizers will basically make these sequences available. These sequences will all be of proteins that have been crystallized but are embargoed. So the people that participate in the competition can't see the structure, they only get the sequence. And so the point is you have to use this sequence and then develop a program to simply use the sequence to predict what the protein structure will be like. And the protein structure is written to this type of PDB file. So for each amino acid over there, methionine, lysine, glutamic acid, for instance. So for methionine, for each of the different atoms constituting the amino acid, you have to calculate and assign an X, Y, and Z value in Euclidean space to each of those atoms. And at the end of the day, when the competition is done, what the organizers will do is they'll take all the submissions, and then by that time the embargo would have been lifted on the structures and compared to the actual structure that's been deposited into PDB. And compared, and if you basically predict a protein structure that's very close to the real protein structure, you get a good mark. And what happened is that if you look over here, so this is the result from CASP 40, which was in 2020, and this is basically just an arrangement of the scores of the various groups that participated. And you'll see the one that absolutely stands out is AlphaFold 2, and then also David Baker's programs, development suites that he used to predict the structures. Okay, so I think I just want to quickly run through this. I'm not going to spend a lot of time on this so that there's some appreciation of what's happening in the background in a program such as AlphaFold 2. So over here, this is just a very simplistic representation of a feed-forward neural network. It's not a complex network. And basically, you have an input layer over here. You have data coming in. So each of these little circles or nodes, I1, I2, I3, etc., have a certain value. And then these values are combined. So you basically multiply the value of I1 with a weight. And then you add to that the value of I2 multiplied by this weight, plus I3 multiplied by this weight. And then the sum of all the different products basically is the value that you write in J1. And then you do the same for J2, basically from these various inputs, and then the same at the next level. So the point is you have some information coming in, you have some weights that are assigned, and then over here you have an output layer. So I've just chose 3. There's no reason why it should only be 3. It can be any number that you choose. And information comes out. And what happens is that when you train this neural net, you basically compare the output of the neural net that is produced with the expected value. And the difference between that gives you an error. And what you can do, the idea is to minimize the error, and you do that by training. So basically you run multiple hundreds of thousands of examples through this neural net. And then I'm not going to go into this, but by a procedure of backwards propagation, you readjust these weights so that after it's been successfully trained, if you input a certain series of values over here, you get the values that are outputted by the neural net. That is what you expect it to be. So this is the basic phenomenon, or basic idea rather, philosophy behind AlphaFold. So there's another level of complication that I would like to mention. I'm not going to go into this at all, and that is the use of self-attention mechanisms. So basically, what happens is you have information coming in, and the proximity. So in a natural language model, proximity of specific words is important. So what you're trying to do is you have to apply an impact score to that proximity of specific words as a subject and a verb, etc. So that in terms of protein structure, that would be the spatial proximity of specific groupings of amino acids. So there are many of these attention heads that are used in the entire algorithm itself in AlphaFold. And here is a very simplistic representation of the AlphaFold 2 algorithm. So you start off with a protein sequence, then the algorithm will go and it will query lots of different genetic databases and find related sequences. And from that related sequence, it will basically set up a multiple alignment matrix. And this provides some information about the evolutionary history of the protein. Then after training of this, and obviously when you use it, it has been trained, there's also information in terms of the distances between specific residues. And so this step here is part of what I refer to as the EVA format. And essentially, it is a combination of the evolutionary history of the amino acids and then the pairwise distances between specific amino acids. And the output from this is fed back into the EVA format multiple times. And eventually, it's passed to a structure module, and the structure module makes sure that there are not any or disallowed clashes or anything like that that's present in the model. So it basically produces several models and it gives confidence scores to each. And you can basically get to the model with the highest confidence and take that as the predicted structure by AlphaFold 2. So just as an example, over here I did this. So over here is the crystal structure of pyruvate kinase. And then I fed the sequence of pyruvate kinase into AlphaFold 2. And this is the structure that is predicted by AlphaFold 2. And then if I superimpose the crystal structure with the predicted structure over here, retaining the colors, you see you end up with a superposition with an alpha to alpha root mean square deviation of less than 1 angstrom. So AlphaFold 2 is pretty accurate in terms of predicting what this structure should look like based simply on its sequence. Okay, so something else that John Jumper and Demis Hassabis did is earlier this year they published another paper in Nature, May of this year, accurate structure prediction of biomolecular interactions with AlphaFold 3. So it's a new version of AlphaFold. And over here, basically reading, so far greater accuracy for protein and ligand interactions compared with state-of-the-art docking tools, much higher accuracy for protein nucleic acid interactions compared with nucleic acid-specific predictors, and substantially higher antibody antigen prediction accuracy. So basically, this extends the ability of predicting structures and also does so with a much higher accuracy. Okay, so what would one use if you applied Alpha 2 or AlphaFold 3? So one of the things would be drug discovery. So you basically look at, so especially now with AlphaFold 3, which has this capability, look at specific substrates or ligands and binding to proteins that could influence the ability of the functionality of that protein. You can look at disease impact of genetic variants. You have a gene, and you know that in that gene there's a specific modification, and you know with that modification that would cause a difference in a amino acid. So you basically take that sequence with a modified amino acid, and you predict the structure of the protein, and then you can get some insight into the possible impact of that genetic variation. You can look at protein-protein interactions. So over here, I'm just showing one sort of hydrophobic surface plus a cartoon representation of two proteins interacting, showing that one can sort of study these types of interactions as well with AlphaFold 2 and 3. And then lastly, is basically the design of novel proteins. So I'm referring here to protein X. So for instance, what if you basically looked at an enzyme and you thought that if you actually shifted a positive charge slightly in this direction by introducing some amino acids, basic amino acids, to basically carry that positive charge in this area, that would improve the efficacy of the enzyme, the specificity of the enzyme, or designing an enzyme that can actually perform a structure that doesn't yet exist. So this is exactly what the other person in the Nobel Prize did, and this is David Baker. So I'm doing him injustice by just pulling out this one paper, which is from Science, on protein in PNN. So he did a lot of protein design work prior to this paper, but this paper has had a significant impact on protein design, which is one of the reasons why I'm pulling it out. And what this paper does, it basically approaches the problem from the other end. So instead of giving a sequence, looking at the structure, it's given a structure and then asking, what is the sequence, okay? And so, and once again, I'm not going to delve deeply into the algorithm here, but essentially you have a protein structure that's represented. You look at the different alpha carbon atoms, the imino, nitrogen, carbon, etc. And you look at the pairwise distances between the various atoms in the structure, basically making use of a backbone encoder, passing this on to a sequence decoder, and ending up with a sequence that is predicted will fall into the structure that you basically passed to protein MPNN. So what are the applications of this? So this is very diverse as well. De novo protein design, de novo protein design, therapeutic protein engineering, if you want to basically change or fix a protein, enzyme engineering, if you want to make an enzyme that has a specific change in efficacy, protein stability optimization, binding affinity optimization for protein-protein interactions, vaccine development, research basic science, asking fundamental questions about protein structures, protein folding pathways, etc., and drug discovery and development. And then in my last slide, I just want to sort of point this out in an era of synthetic biology. So those two spheres over here, those are the oxygen atoms of water molecules. This is simply glucose that has been phosphorylated at its carbon 1 position. And then this area, shaded area around it, is part of an electrostatic surface of an enzyme. And the point that I want to make with this is that looking at a specific substrate, we're starting to knock on the door of the ability, given the tools that was developed by these three gentlemen, for which this Nobel Prize was awarded, we're starting to knock on the door of looking at these substrates, thinking about a chemical reaction that we want to take place or want to catalyze, thinking about what would be required in terms of the distribution of electron densities in the substrate, that would be related to the distribution of charges in the vicinity of the substrate, thinking about what the conformation of the substrate would be to resemble the transition state of the product. So these all basically indicate or basically suggest, given the tools such as protein MN, message passing NN, that one can go in and you can basically design the structure with the distribution of charges in the enzyme to basically catalyze a reaction that you design here. So we're not quite there, but as I say, we're starting to knock on the door of that type of ability. And when we can do this, then we enter an era where we can basically design enzymes. We can easily synthesize the genes encoding those enzymes, clone that into bacteria, express in bacteria, isolate the proteins, and we can actually make bacterial or other cells that will catalyze specific metabolic steps that are not found in nature now. So we can actually start thinking about designing new metabolic paths and having synthetic metabolic pathways in organisms. So this is where we sort of heading in terms of the capabilities that comes from this Nobel Prize. Okay, I think that's full of halt. Thank you very much.
So, so if you can ask the speakers, Johan and Kristian, to come to the front, and then the floor is open for speakers for questions from anybody. Vida's looking online. Vida. So if there's questions online, please just speak up as well. Yeah, so the floor is open. Any questions to any of the speakers? Yes. Agree.
Yeah, so I've got two questions. The first one's to Prof. Burger. Thank you for a great presentation. I think my question is to be what controls the expression of microRNAs? So there's a few parts to this. Are all microRNAs at all times abundant as present? And if not, what controls their expression? What feeds signals to say, I now need to down-regulate the mRNA of whatever, because it's already been transcribed?
Margaret, simple answer, I don't know. No, so the interesting thing is there's an anti-correlation between microRNA expression and their target, right? So, but they are so tiny, and I see where you're going and what you're hinting, but they do not have proper structured promoter sequences. You can imagine a normal promoter sequence and then this tiny little thing. So when and how the initiation of transcription, I don't know. What I do know is they also transcribe by RNA polymerase II, just like normal genes. And whether they constitutively expressed, that would be my guess, but I honestly just don't know for sure. Sorry, was there a second question?
No, I didn't really ask the question, but the whole concept of the fact that RNA is being expressed by in a more diurnal manner, which we never really realized that it makes a whole lot of sense. I'm wondering if microRNAs might be under a same kind of levels of influence.
Yeah, the interesting thing of all the small RNA species that's been discovered, and there are lots of them now, they are the only ones that are genome encoded. The other ones are all triggered by aberrant RNAs of some sort. So for small interfering RNAs, for instance, are triggered by the presence of double-strand RNA, which is an intermediate for viral replication and stuff like that. So they are unique in that they are encoded, and that is also illustration of their evolutionary importance, obviously. Yeah.
Other questions?
For the, there is a bit of controversy for the first one that it is in the field of physics. It's not eager to classify this rather in the field of mathematics, say the fields may involved in probably involved in age. Would you agree that it is the physics Nobel Prize equivalent, or would you withdraw the that's fine? The generation function mathematics.
So I wouldn't know how to answer. So the way, I see it, certainly there's very much physics inspiration into effects that physicists are familiar with in generating at least the seed of some of the ideas. So definitely I agree, perhaps, definitely many aspects of refined algorithms might not necessarily always be physics, but I think that also, as we've discovered in repeated Nobel Prize lectures, the boundaries between disciplines are not very clear. Actually, there's considerable overlap, and any field as it evolves probably moves around to spread the kind of cloud. So I think it's sometimes difficult, and I think if we look at the history of Nobel Prizes, on quite a few occasions, one would have not been able to pinpoint exactly this chemistry or this physiology, and there's a real continuum. So
very adequate answer to that.
Yes, Willem.
Well, balance it out. A question for you. Yeah, it's not really my area of expertise, but the AlphaFold principle still makes use of many structures of the PDB. Am I right? How close are we? Is it a step in the to determine functions structure without having to refer back to where one could actually look at that sequence, come up with a system that is able to work out what it's going to fold, almost in an evolutionary sense, without referring. I'm just trying to. I get there's a lot of wonderful crystal structures, and that's the one part. The other, are we getting to crystal structure or predictions of structures that we have no idea what they actually do? Or just give us an idea of what.
So the first part of the question. So for many years, the approach has been to try and use physical chemistry to calculate the various types of interactions between parts of the proteins, and then trying to fold the protein. And so you might have seen years ago, there used to be something that used a screensaver called basically fold at home, which was basically a parallelized approach to use computers in the off time to try and fold proteins. And each basically individual program would try something else, calculate the interactions, and then get to a point of the lowest minimum where it wasn't possible to jump to anything lower with this sort of temperature type of approach that we heard of in the first talk. So that has been the approach for a long time. And the big step forward here was saying, well, let's stop doing it that way. Let's see what we know. So looking at the PDB database with hundreds of thousands of different crystal structures, let's see, can we learn anything by looking at the structures and the arrangements of the amino acids in space, in these various structures, to actually train a neural net, and then use that neural net to apply to a sequence to try and derive a structure. And it seems to be very, very successful to do that. So I don't think that our understanding at this point in terms of the physical interactions at that distance, limited distance, is sufficient to actually allow us to properly predict what the final structure will look like. The second part of the question is, have we ever identified a crystal or structure of a protein that we looked at and we say, boy, we have no idea what this does. So the thing is, there are various types of things. So you can obviously, if you have the crystal structure, you have the sequence, because you can actually derive the sequence from the crystal structure, but you would have overexpressed the protein to get the crystal in the first instance. So you have the protein sequence, and you can basically search it against the database, the non-redundant database of all protein sequences that we've ever sequenced, including all protein sequences predicted from all genomes and genes in sequences that we've ever sequenced. So we have this enormous database that we can search and see, do we have anything that looks like the sequence? Apart from that, you can take the crystal structure of the protein, and there's a way in which you can do a structure search. So basically go through and basically compare to all the various structures and see which one fits best. And the combination of having a sequence that is most homologous, as well as a structure that fits best, gives you a very, very good chance of at least having some guess at what the likely structure of this protein is. I've never seen something that says, well, here's this protein, we have no idea what it's doing. I've not encountered that. But just because I haven't encountered it doesn't mean it doesn't exist on this one.
So on the question of protein folding and perhaps the idea of designing proteins. Yeah. So these algorithms just find the folded structure, but then one of the things at least at some stages, how quickly can that protein fold, or are there sort of dynamical influences to the folder structure? Does that play a part in any of the current algorithms?
That's a very good question. So basically, that's actually a little bit of caveat, and I glossed over. So you can get a sequence and you can get the predicted final structure, but there's no guarantee that that final structure, that may be the correct final structure, but there's no guarantee that that final structure is attainable via any folding pathway. So as you would know, I mean, you can sort of attempt to represent a folding pathway as some type of funnel, energy funnel, and you move down to the lowest energy at the bottom, and there's a specific pathway that you follow to get to that point. So the issue is, it may be that the flip sizes on that energy landscape for that particular protein sequence is such that it gets to a point that even if you have some type of environmental thermal contribution, that it can never shake loose of a misfolded sort of section. And that has happened. So we have seen, in my group, we actually developed software that did something very similar to protein MPNN. And we could synthesize the proteins with a different sequence, with the idea that one can actually modify the protein to modify specific amino acids in the protein to tweak the functionality thereof. And what happens is that, yes, we can make the gene, and we can never see the protein itself, even though the gene is active. So the RNA comes off, but the RNA never goes to the protein. And the reason for that is the protein is synthesized, but it misfolds and is then very rapidly removed by the cell. So just because AlphaFold will predict a structure, or AlphaFold 3 will predict a structure for a protein, doesn't mean that that protein will assume that final structure, and that's attainable in terms of the folding part.
Thank you. It's been the last
But you also suggest going kind of looking into the future, what we're able to do now, and we think of reactions like the electron transport chain, ATP synthesis, those kind of things. How do you see it if we fast forward 15 years or so, and the mitochondrial dysfunction and
Right. So I think that the first thing that I should perhaps say about that is that evolution is exceptionally efficient. So basically, the whole system that you have information there, the information can be tweaked, that tweaks the system. The system is tested in terms of its survivability in the environment. If it's better than the previous model, it will basically have to replicate more, there will be more copies of that particular system, and it will eventually take over the population. So the whole concept of evolution, even if you apply it to computer software in terms of genetic algorithms, is also extremely efficient. To teach things, for instance, just a simple game of Pong, where you have two bats moving and you have a ball basically bouncing, and you use a genetic algorithm to evolve the system to know how fast the bat should move depending on the angle and speed of the ball. And very, very quickly with the genetic algorithm, it sort of solved that problem. So given, was it 4.5 billion years of evolution, I think a lot of the systems that we have now are really, really efficient. And that one makes the mistake in thinking, well, we are complex and bacterial cells are evolutionary ancient. They've been evolving with us for the past hundreds of millions of years. So from the earliest bacterial cells, which probably dates back to about 3.5 billion years ago. So that is part of the answer. The other part of the answer is, if you look at systems like electron transport, there's no reason why there aren't additional things that you can think of, that you can tweak and change. And especially if you start looking at an organism as a graph, a mathematical graph, so nodes and edges connecting the various nodes. And the nodes can either be the reactants in the chemistry, or they can be functional groupings in the cell. So nodes that are highly connected in this graph are not nodes that you can go and sort of easily tweak because they may negatively impact a lot of other things. But there may be nodes on the outside part of this graph that have few connections that you can easily go in and you can tweak. And so if you can identify those nodes in a functional description, say, of electron transport, you can go in and modify that. And there's no reason why in sort of a final analysis, and we're not nearly there in terms of our biochemistry and our understanding, but in the final analysis, going and designing something that might be sufficient, that might be significantly more efficient than electron transport, because evolution basically maneuvers it often into cul-de-sacs. So once it's sort of developed this network of connected nodes, it's very difficult to break a lot of them, just break them away and sort of repopulate with other nodes. Because if you do that, you break the entire system. So once you've established something, it's very difficult to go back and fix things to go forward again. So that's the one drawback of evolution. But I think sort of given a very, very long perspective, I see no reason why not only with electron transport, but with photosynthesis and a lot of other metabolic reactions, why we can't design our own custom enzymes that will catalyze reactions that are not found in nature now.
Okay. Are there any final questions before we move on? I don't see anything. Yes, final question. Right.
Chemistry notes and function leading to the neural network, and we see that applied in the end of the process. This whole idea behind the physics Nobel Prizes, I guess the big search in ChatGPT and things like this. I guess maybe open-ended to any of the speakers. Do you think philosophically, maybe, I don't know if you want to call it physics on this round, this tells us something about the energy landscape that is inherent to so many things, from protein structure to language to many things, that this principle holds on so many different levels?
It's a difficult question, Kristian.
Trying to go for these things, so if you judge it, action.
I think I can comment that there's some really elegant and simple math actually behind a lot of these models. It's not complicated stuff. It's looking at distances between vectors and things like that, which we teach our first years. That's the underlying maths. But it gets a lot more complicated on this level.
I would argue about that. So I mean, the sort of physical laws of the universe. That's all the universe can represent are the physical laws. And so anything that happens in the universe will have to comply with those physical laws. At this point, as far as we know, in terms of the current physics, you can't go faster than the speed of light. If you have two objects with certain mass, there's some what we describe as a force, but I think physicists will more formally describe that as curvature in space and movement along the space-time curvature, as opposed to like a force between objects. But so there are lots of laws that are, and these laws are applied to living systems as well. And I think one of the things that I see one of my students here that are actually working on aspects of this that is relevant, and that one of the big things with living systems, and that's also an issue of physics nowadays, is the development of complexity. So we live in a universe that where we see the observation is. So we have a mathematical description thereof, but we don't know what the underlying reason is, that there's always this movement towards basically an increase in entropy. And so if you have an increase in entropy, how can you have highly ordered systems developing? And if you have a highly ordered system developing, how can you have a highly ordered system becoming more and more complex and not simpler? So isn't that sort of going the wrong direction? And I mean, the simple answer to that is, well, look at the system as a whole. And so yes, the entropy of the whole system will increase, and there may be sort of local areas decreasing, but as long as the entire system stays the same or increases, then that doesn't basically break the rules of the universe. So there's nothing in terms of living systems or biology that we see that is contrary to fundamental laws of the universe. That, I don't know if that addresses the concern. Thanks.
Right. If there's nothing further, then I think all that remains for me is to thank our speakers in the very traditional Stellenbosch way.
Thanks very much. Appreciate it.
And Kristian. Right, okay. And then there was an attendance list that circulated around. So if you can make sure that you sign it, I'll appreciate it. Just for us to keep track and tell the sponsors that there were people here. We didn't just eat their food and drink the wine. And then I invite all of you up. You can follow some of our postgraduate students. We're up in the electricity lab. There's some snacks and some drinks and some wine. So hope to see you all of there to get something. Thank you all for coming, and to those online. All right. Dank je. Ja, nee, ik denk zo. Sharing.